Welcome, everyone, to our next topic, which is genetics. This topic builds on what we covered previously, topics on DNA, the code of life, mouths, and reproduction. I trust that you still recall the key content from those sections. Right, we are going to explore genetics in two parts, part one and part two. Today, we're looking into part two. Right, let me put this on the presentation. Okay, this is the presentation outline. I'm going to first introduce the topic genetics to you. Then we'll be unpacking concepts about inheritance, then tackle Mendel's laws of dominance, segregation, and independent assortment. We're also going to engage in a number of genetic problems, such as monohybrid co-dominance and incomplete dominance. Right? What is genetics? Genetics is the science of inheritance and studies the principle of heredity and variation. Gregor Mendel is called the father of genetics. He performed quite a number of experiments using garden pea plants, also called Pisum sativum. Now, why did he use the garden pea plant? It's because the flowers of this plant is bisexual, and this plant is self-pollinating, and he was able to manipulate this plant by allowing the so-called cross-pollination to take place. Mendel introduced three laws: the law of segregation, law of independent assortment, and law of dominance. What is the law of dominance? This law is indicated when two homozygous organisms with contrasting characteristics are crossed, and where all individuals of the first filial, abbreviated as F1 generation, will display the dominant trait. The law of segregation is indicated when an organism possesses two factors which separate or segregate so that each gamete contains only one of these factors. The last law of independent assortment is the law that indicates that various factors controlling the different characteristics are separate entities, not influencing each other in any way and sorting themselves out independently during gamete formation. Well, remember what I said in my introduction? I said this topic is linked to our previous topics. Here are concepts that we have learned previously. Remember, each and every subject has got its own language. Hence, I prefer to introduce concepts, which is the language of a particular subject. Well, we have learned about what chromatin network is, also chromosomes, gene, allele, and locus. But today, here is a different and new concept about genetics. It is called the genotype. What is the genotype? This is the total genetic composition of an organism. Simply means all genes that one possesses. We inherit genes from our parents, the father and the mother. So a total of genes that you have inherited from your parents are called the genotype. And the phenotype refers to the external or physical appearance, the outward appearance. Let's say, for an example, your father is tall and your mother is short. And you are tall. When we say you are tall, we're looking at the outward, the external appearance, which is determined by genes that you possess. What is a dominant allele? This is an allele that masks or suppresses the expression of the allele partner. Remember, an allele consists of two genes from mother and father. So when one of the genes from this allele suppresses an expression of another partner, we say it's a dominant allele. Okay? And what is the recessive allele? It is a direct opposite. It is that suppressed gene from the pair of genes, which is an allele, the one that is not visible. Can I take you back to what I said initially about you being tall and have inherited the tallness from your father, who is tall, and your mother, who is short? Now, when we talk dominance, we're referring to that gene of tallness that you have inherited from your father. It is dominant. It suppresses the gene of shortness that you inherited from your mother. Now, the recessive gene, it is that suppressed gene of shortness. It is not visible externally. We don't see you as a short person. You are tall, but the gene is there. Right, and we also talk about multiple alleles, which refer to having more than two possible alleles for one gene locus. We talked about gene mutation in the previous topics. Then this allele or pair of genes can be homozygous or heterozygous. What is a homozygous allele? The word homo firstly means same. So these are alleles that control a single trait and are alike. Remember, we are going to talk more about these homozygous and heterozygous alleles. And in genetics, we use letters of alphabets to indicate an allele, a pair of genes. So when we say this allele is homozygous, in using these letters of alphabets, we'll either use two capital T's or two small t's. They are homo, they are the same. Conversely, a heterozygous allele is the one that has got different genes, meaning unlike the homozygous, when we either have two capital letters or two small letters, in a heterozygous allele, which the word hetero means different, we will have capital letter and a small letter. Then what is incomplete dominance? In an incomplete dominance, no one allele is dominant over the other. You are going to learn more about incomplete dominance and the complete dominance in the next slides. So complete dominance is when one allele is dominant over the other. The other one is recessive. Right. As indicated, we're looking at part one of genetics. We're focusing more on monohybrid crosses. These crosses are used to understand all the three laws by Gregor Mendel. The word mono means one. So in the experiments that Gregor Mendel performed, he used one pair of a trait to determine the possible inheritance in the offspring. So the word mono simply means he used one characteristic. It could be the height, it could be the color. So let's use an example of a general trait like the flower color. Remember what I said? We are going to use letters of alphabet to show dominance and recessiveness, meaning the gene that suppresses another gene and the gene that is being suppressed. Whenever you see a capital letter, you must know this is a dominant trait, and a small letter, it's a recessive trait. So we are going to use here one trait, which is the purple color, a plant having flowers that are purple, which is dominant over another plant having flowers which are white. Right? Let me show you the structure here to show the flower purple from one plant and the flower which is white. Remember what I said? We are using letters of alphabet, capital B for purple, to indicate it is dominant. And we're using the letter of alphabet for white, small letter B, which indicates recessiveness. It is being suppressed. Now look at this Caesar here. You might get this kind of a diagram in your examination. Kindly note that this Caesar is used to remove anthers. Remember I said Gregor Mendel used a plant that is bisexual that can self-pollinate. Now, because he wanted to show the inheritance from two different plants, he had to remove the anthers to prevent what we call self-pollination. So the anthers are being removed from this plant so that anthers from another plant can be used to pollinate this plant. So in here, it means pollen grain is now being delivered from another plant to another plant. We refer to this process as cross-pollination. After cross-pollination, the seeds are here, and when planted, we find that the first offspring, abbreviated as F1, first filial generation, are all purple, because we said color purple is dominant over color white. Right. Know that you will be exposed to these kind of activities, and you have to use a table to indicate the crosses. Remember we said the parents, we have got two plants, the other with purple flowers, another one with white flowers. So this is parent one, P1, parent one. And phenotype, the outward appearance of these flowers, the other one is purple and the other is white. And what is the genotype, the genetic makeup? Remember, genes are always in pairs, it's an allele. For the purple color, we said it is dominant. It is represented by capital B. And the white is recessive. represented by small letter B. Every time you are asked to do crosses of a recessive characteristic or trait, use small letter. But when you are expected to show dominance, use capital letter. Meiosis will take place during the formation of pollen grain, and gametes, which is pollen, are going to be formed, which are haploid, meaning having half the number of chromosomes, and fertilization will take place. The same information here can be shown here in the form of a table. What you need to do, you show the allele or the genotype for purple, which is 2 capital B, they are here. Capital B, capital B. And for the white, small letter B's times 2, here is small letter B, small letter B. And how do you do the cross? Small letter times capital letter is capital small letter crossed by capital letter, small letter. Then you come down here, small letter B, crossed by capital letter B, it will be capital letter, small letter B, crossed by capital letter B, it will be capital letter, small letter. So these are the offspring. All these 4 represent the first filial generation. And then, what is the genotype? All of them have capital letter B and small letter b. Because these pair of genes and allele are different, we call them heterozygous offsprings. The word hetero means they are different. And what is the phenotype? Since they contain a capital letter that shows dominance, they are all purple. Okay, let's look at another example. This time we are crossing heterozygous by another heterozygous. Remember what I said? Heterozygous means different. So here are these 2 plants that we are talking about. Is parent 1. The phenotype is purple and purple this time. But then let's look at the genotype. The genotype is different because it's capital B and small letter b. So it's heterozygous. We're using the same method. Meiosis will take place, gametes will be formed, and fertilization will be formed if we transfer the same information to this table. Remember what we did? These are the allele or the genotype of each and every plant. It's capital letter B, small letter, capital letter B, small letter. Remember we said heterozygous crossed by heterozygous. So here are the offsprings. The so-called F1 generation. The genotype, we have 2 capital letter B's. We also have 2 capital letter B and small letter, and 1 out of 4 small letter B's. So we have homozygous, heterozygous, and homozygous. And the phenotype is 75% purple and 25% white. Another example of a monohybrid cross. This time, it's a cross between homozygous dominant and heterozygous. Homozygous, these are the 2 B's. Both of them are capital letters. It's homozygous. Heterozygous, this is capital letter B and small letter b. And these are the offspring, the first filial generation. What is the genotype? Two of these offspring have the same genotype. They both have two capital letter B's, and another two also have the same genotype, which is capital letter B and small letter b. But in as far as the phenotype is concerned, they are all purple. This is the last example. We are crossing a plant with homozygous recessive by the one which is heterozygous. Homozygous recessive is small. Remember, I said whenever you see the word recessive, think about using small letter. They are homozygous, they are the same. Heterozygous, it's capital B and small b because they are different. When you cross using the same method, the offspring will be these. The genotype, 2 out of 4 will have capital B and small b, which is heterozygous, and also another 2 will have 2 small letter b's, which are homozygous. So these are examples of monohybrid cross. Right? Fine. Now we are talking about incomplete dominance and codominance. What is an incomplete dominance? This is a dominance or a characteristic that happens when two true breeding parents are crossed and they produce an intermediate offspring, meaning there is a lack of dominance in either of the two different alleles. Okay, here is an example. Here is a plant with red flowers, and there's capital letter. Whenever you see the word capital, you know what it means. And there's another plant with white flowers, presented as small letter R. Okay, instead of us having dominance here, complete dominance of the offspring, we have incomplete dominance. It's an intermediate offspring. It is pink in color. So the red and the white produce a pink color. This is what we call incomplete dominance. But codominance, as represented by this diagram of the bull and the cow, the bull, which is cream white in color, and the cow, which is red. If mating takes place and there are calves that are born out of the two, both calves have the color or the allele of both plants. The calves have the color of two parents, the red and the cream. So not the red color nor the white color is dominant. The calves have colors of both parents, and we refer to this as codominance. Codominance can be used in blood groups. You can be asked a question where you need to do this genetic process using blood group. There are 4 different blood groups. We've got A, two A's with different genotypes, two B's with different genotypes, and we have AB and O blood groups. So they can be used to show codominance. For example, if you are to cross an organism with this blood group with another organism with this blood group A and blood group B, know that the offspring or the children will have both alleles of both parents. It will be A capital and B capital. So this is an indication of what codominance means. It can also be applied in flowers, where there's a red flower that is being cross-pollinated with a white flower, and we find offspring or the new plant from these two plants having two colors, the white and the red. It is codominance. Right. I hope you were with me. I want us to do a little exercise, because our next topic will be about a dihybrid cross. I just want to be sure that you have mastered the monohybrid cross. Fine. What do I want you to do? I want you to make a cross between two individuals. The one has got brown color of the hair, and the other one blondie color of the hair. Note that the one with brown color, the trait is dominant. It is capital B, and the blondie color is small letter b. But here is a very important instruction. What do you need to do? You are going to cross homozygous dominant by homozygous recessive. Please think back what I said about the word homo. It means the same. It means the pair of genes or an allele that you're going to use in the table must be the same for a dominant trait and must be the same for a recessive trait. It's a brown color that is dominant and a blondie color of the hair that is recessive. Use this table, and this is how you need to do or to answer questions in the examination. You need this table. First, indicate the parent. Are you doing it? Indicate the parent. Let me remind you, one parent has got brown color hair and the other parent a blondie color hair. Write the phenotype, write the genotype of parents, and indicate the gametes after meiosis. Are you able to locate the gametes? What are the gametes? And fertilization. Right. You are going to take those alleles, pair of genes, and put them here on this table. This will be for one parent. This will be for the other parent. Quickly do the crosses. Do the crosses quickly. Of course, after doing the crosses, you'll be able to note the genotype and the phenotype percentage. Are you there? Are you struggling? Hope not. Right. This is the marking guideline. You must have indicated parent 1's phenotype is brown and blondie. Brown is capital letter B times 2. Blondie, small letter B times 2. After meiosis, here are the gametes. The gametes need to be transferred here to show fertilization and the formation of the first filial generation, the F1. And here are the gametes. Here is the genotype. This capital letter B and small letter b. All 4 of them are heterozygous. And the phenotype is all of them are brown, since brown is dominant. Okay, thank you so much for listening. Our next part will be genetics part 2. We are going to look into dihybrid cross. Thank you so much.